Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we would like to take a moment and thank our Dr. GPCR Ecosystem partners for their support. Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. We hope that you were able to join us at the Dr. GPCR Symposium on Molecular and Structural Insights in GPCR Activation. If not, you can go back and watch the recorded talks with your premium membership. Also, mark your calendar for the upcoming symposia on September 22nd on the topics of GPCRs as therapeutic targets. If you'd like to be considered as a speaker or have a speaker suggestion, please email us at hello@drgpcr.com. More information can be found in the ecosystem. If you haven't noticed yet, we've added menu buttons at the top of the page for your convenience to help you navigate the ecosystem more effectively. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. This morning, I'm very excited to have with me Lukas Grätz. Hi, Lukas. How are you?
I'm good. Thank you.
Thank you. I'm so happy that we were able to do this, especially that we met in person 2 weeks ago at the G Gordon conference. We were just talking about it before we hit record. So how about we start at the beginning and you introduce yourself?
Yes. So first of all, I want to thank you for inviting me before I miss that, or before I forget that. So about me, I'm Lukas Grätz. I'm from Germany. I grew up in a small town, a small village in the southeast of Germany. And after graduating high school, I studied pharmacy in also university in the southeast of Germany, pretty small one. It's Regensburg, a beautiful city, by the way, so might be worth a visit. And after studying pharmacy, I decided that I want to do a PhD. So I liked the city, so I stayed there. I made my PhD in the medicinal chemistry group for 4 years, no, a bit more than 4 years. And I graduated in 2021. And since June 2021, I'm now a postdoc in the Schulte lab at Karolinska Institute.
Amazing. And when did you first hear about GPCRs? What led you to your postdoc?
That's a very good question.
Considering your training.
Yes. So I also listened to the episode from Hannes Shihada, whom I know as well. And for me, it was quite similar, actually. Mm-hmm. We had the same studies at the same university. And I really liked the lectures of Professor Buschauer, who was then my supervisor, at least for a brief time, in the beginning of my PhD. And I really liked his lectures and his concepts that he explained about GPCRs. But honestly, after I finished my studies, I just wanted to do a PhD. Mm-hmm. I knew that I wanted to stay. So I just looked into the groups that were available, and I thought that this group was the most interesting to me. So I kind of was forced in the beginning a bit into GPCRs, but then I really enjoyed it. And also with the next 4 years, I really enjoyed working with them, working with their complexity. And after my PhD, I wanted to stay with GPCRs again, but maybe switch it up a little bit. So I switched it up as hard as possible, kind of, to go to the class F. And yeah, that's why I'm in Sweden now.
That's amazing. And I was going to ask, what was your PhD project about, considering that, well, I know that you're in Gunnar's lab and you work on class F GPCRs, but you're not easy to work with. Mm-hmm. It's a whole different beast. So tell us a little bit about your PhD project.
My PhD project was a bit of an outsider an outlier in the group, because as I've mentioned, it was a medicinal chemistry group. So most people were synthesizing new compounds and trying to find ligands for some receptors with very high affinity, or making new tracer molecules. And my topic was one of the few which was focused on pharmacology. So my project was about developing bread-based binding assays, because when I started, they became more and more popular in the field of class A GPCRs, and they were also the first publication about the application for other GPCRs. And then it was my task or my project to develop these assays for the receptors that were investigated in our group, mm-hmm. which was sometimes easy, sometimes not so easy, but that was kind of my project.
It's interesting because that means that you were in the right position in that sense, because you were getting molecules and then you were testing them, kind of like the chief tester, and everybody, I guess, had to be nice to you. I'm kidding. I'm kidding.
I also
but think about it.
So
But think about it, you were the gatekeeper when it came to pharmacological data on the molecules in that sense.
Not really, because my technique, we still had radioligand binding. A lot of it still had it in the group. But for me, it was more the other way around, because of course I had kind of a compound catalog in my group. So if I had a very interesting or very, very potent compound in the group, I just asked them, hey, could you make it maybe fluorescent? Could you attach this label? Could you attach that label? And then I was able to test them. So that was more
the other way around.
good opportunity for me, I would say.
Absolutely. I think it's great because it allows, it's a great way to collaborate. You have multiple people working on different topics in the same lab, or different techniques in the same lab, all with the same goal, is to better understand GPCR biology. And then you had access to these tools. You mentioned having a full catalog of molecules, which is actually quite cool. So how did you, you mentioned, switching and going to Karolinska and switching to Class F GPCRs. When you were looking for a postdoc, how did you go about it?
So I graduated right when the pandemic hit, when the pandemic was pretty strong, at least in Germany. And during my PhD, I knew that I wanted to go abroad, but my goal was more to go further abroad, like over the Atlantic, more US or Canada. But then when it was time to make the decision, I thought, okay, that might be a bit too much now, also with the regulations. So I looked for something in Europe. And in this case, Hannes came in handy again, because I knew him before. So I got in contact with him, or we got in contact, and he told me about it, about the projects, about the atmosphere. And then I just contacted Gunnar in this case, and he was open for it. And that's how it began, kind of.
That's great. And you make a great point about, reaching out to someone who who you trust, obviously. And I mean, Hannes is really wonderful. I really enjoyed talking to him, and he's presented a couple of times within the ecosystem as well. So it gave you a great, insight into how the lab might function, what are the projects. You went into the conversation with Gunnar very well prepared. And that's very important.
I would agree. It's always good to know someone who already knows the drill, kind of how it works and how life is in the country and in the lab, about the project.
Was it a big change for you to go from Germany to Sweden?
Well, of course, it was a big change. Going abroad alone, like being in a new country, all the organizational stuff that has to be done in the beginning, it was a big step. But I think Sweden is comparatively easy for Germans, at least, because they are not too far away. Also, both in terms of distance, but also in terms of mentality. They are a bit different than Germans, but at least it's not the complete opposite. So it was still okay.
That's great. The reason I was asking is because sometimes, people are thinking about maybe, oh, I should do a postdoc abroad, and they don't know how to go about it. And it might be difficult. And we've had many guests who went from, India or Europe to the US. And I always ask about how was it happening, and what did you learn during that adaptive phase?
Yeah, as I've said, it was maybe not the biggest culture shock for me, but of course I learned a lot also about the Swedish system, of course, which can be very nice. It also can be pretty bad in some other aspects. But I learned a lot about this, and also just being responsible for yourself. There is, of course, you get help if you want to, or if you need it, also from other members in the group who are more experienced with the Swedish lifestyle and the Swedish system. But you're kind of on your own in the beginning. I learned a lot during that process, definitely.
That's great. And I think that's how you grow, is you get out of your comfort zone and you go and do something completely different. And, I think I was talking to someone on the podcast. I can't remember. Maybe I was talking to somebody outside of the podcast, but they mentioned that when they went away for their PhD or their postdoc abroad, that was the first time they had to do their laundry.
That's a rough progression then.
And I thought, wow, okay, that's actually, I guess there is the time where you have to learn to do that. And laundry maybe is a small step, but when you're on your own in a foreign country, that's when you learn a lot.
I agree. I totally agree.
And it's painful, but it's great because after that you come out, more experienced, more grown, even as an adult.
I hope so, and I would say so now. Yes.
No, I think so too. I think no matter the experience, it just gives you that extra, that extra push. It shows you that you can do it. You may not realize it in the moment, but with time, it just gets better. All right, let's talk a little bit about your favorite GPCRs, because now you mentioned working in a lab where you had to work with multiple Class A GPCRs, and then you switched to Class F GPCRs. Tell us a little bit more about your favorite ones or your favorite receptor.
Right now, I almost have to say it's frizzles.
Yeah, of course.
No, but right now, in the moment, I would actually say it's frizzles. I don't have a particular one in mind. But I think that frizzles in general are a very interesting family, and there's a lot to explore, and it can be very rewarding if you find something, because it's, as I've said, it's more like unexplored territory. There are not many people working on them, and they're kind of a big question mark how they function, how they work, where they are expressed, and so on. It's very tricky. So I think I would go for frizzles now.
Especially, I saw your presentation at the Gordon conference, and I thought it was really excellent. That you're one of the experts on the frizzled family. When you joined the lab, obviously it was clear that you would be working on frizzled receptors, but how did you go about building out your project?
So in the beginning, I applied for funding. Or I got someone to pay my wages and to pay me. So in the beginning, it was also about this project. I had to deal with this project, and I had to get started somehow. And I started in summer, which is always a bit tricky in Sweden, because in Sweden, usually after midsummer, which was a few days ago now, most people go on vacation.
Oh.
For some time, so I was not completely alone, but I think we were 2 or 3 persons in the lab at the time. So I had some time to explore a bit. But then from there, I mean, it worked in the beginning, but then I also started targeting other projects, or maybe addressing other questions that maybe came to my mind, because it's Gunnar leaves this kind of open to us. Of course, we have to work on the project that we have, but he's very open for new ideas, and that's kind of what I did from then, like starting new ideas. If I had something, if I read something, I started with that. So I still have my project from the beginning, but it's kind of evolution, I would say. It's developed.
That's great, because I think it's a great strategy, because you get to work on something that has been proposed, but at the same time as you develop, you can take the project in so many directions that it gives you the opportunity to, make new discoveries, and more brains make for more project ideas, more potential results. So tell us a little bit about frizzled receptors. I think beside you and Gunnar, you're the only two, and Hannes, obviously, the only 3 guests we've had from the same lab working on frizzled. And then we had oh goodness, what's his name? Oh, my brain is not functioning this morning. We had Ben Myers working on also Class F, but on smoothened receptors. What do frizzleds eat in the winter? What are the main questions that you're interested in answering?
Yes, I can try at least to start to explain them. So frizzles belong to the class F of GPCRs, which is really a comparatively small one. The class F consists of 11 members in total. One of them, as you said, is smoothened, which is involved in hedgehog signaling. But then the other 10 are all frizzles. So the numbering is very easy, or the naming, it's frizzled 1 to 10. And they are absolutely important for development. Actually, all class F receptors, smooth and included, are very important for development. So to keep everything in bounds, kind of, the development works as it should. But it still is very important or the signaling pathway, which is the Wnt signaling pathway, is also very important in adults. So it's not only important in development, but it's very important for us now as well. Kind of keeps everything in bounds again. So it's very important for stem cell function, for tissue homeostasis, especially, of course, in the tissues or the types of tissues which are more proliferative, like in the intestinal crypt. That's always kind of the prime example where Wnt signaling is important, or Wnt signaling gradient. Mm-hmm. And of course, if you think one step ahead again, if you look at it from a pathophysiological perspective, if this tightly regulated system gets out of bounds or gets out of balance, then this leads to, or can lead to, various malignancies. And the most prominent one is cancer. And that's why we are also interested in them, to target them therapeutically, or to address them with drugs. And there are still quite a lot of drug discovery efforts going into frizzles now.
What is the best drug modality, you think? Is it a small molecule? Is it an antibody? And you don't, I mean, you can just speculate. I don't have the answer. Sometimes I feel very uncomfortable when people ask me to speculate, but I do have opinions. Yes. So, and it's totally your opinion. What would that look like if the receptors that you're working on from the frizzled family, pretty sure they have some role in malignancies? What would be the best way to block them? Or
that is something that really depends on whom you ask, mm-hmm. and also on the type of disease, or on the context, kind of.
Yeah.
So there were several papers which use antibody conjugates, so bispecific ligands, which kind of bridge frizzles and co-receptors. Also, just recently, there was a very interesting paper about regeneration in the lung. So if you go more into regenerative medicine, I would say you definitely need an agonist. But agonists, of course, are very tricky because you have 10 frizzles, and you kind of want to selectively stimulate them, which can be tough. There were some papers which were successful in that, but that's based on antibodies. But at least our lab is more looking into small molecules. So I think small molecules they are not always superior, but maybe a bit easier to handle.
Yeah.
Especially if it's towards drug delivery and stuff like that. It should not rule out that antibodies it's not possible to deliver antibodies to the right place. I mean, there are several drugs on the market which already succeed in doing that. But right now, I would say for modulation, a small molecule is better. But that's very speculative, and I don't want to annoy anyone.
No, absolutely. I mean, it was just the question was really about your opinion. There's pros and cons in general for small molecules versus antibodies. And I think it's very context dependent. And it's the difficulty with either of those is really hitting selectively and potently enough the receptor.
Definitely.
Independently, whether it's a class F or any other receptor in general. So I think it's really awesome. I forgot to ask you, because you mentioned, being in medicinal chemistry and starting out in chemistry. How did you switch to biology or to pharmacology? Where did that come from? You're not the first guest who started out as a chemist.
Yes. So I'm a pharmacist. So I was not predetermined, kind of. I was not a chemist, or not tending towards chemistry or pharmacology. Mm-hmm. When I started in the group, I knew that now I will be working on GPCRs, but nothing more. So on my first day, when I was still very, very naive, maybe, my professor just told me, hey, what do you want to work on? And I had absolutely no idea. So he just proposed some projects. You could do this, you could do that. He suggested some synthesis projects. And then he added one project which was based on a bread-based binding assay, which sounded very interesting from the beginning. So I just said, okay, I want to have this one. But that was really more like a random choice at the time. So that's how I started with pharmacology or biology, and still stuck with it.
Yeah, well, it was a random choice for me too. I think it's a random choice for a lot of people when you start out a PhD because you just don't know any better. You don't know. And then you get proposed these different projects that somebody thought out, and you just pick the one that you find more appealing at the time. Whether it's a good thing or a bad thing, that's debatable.
Yes. Right now, I would say it's a good thing because I still do not the same thing, but at least I'm still in the same path. So I think it was a good decision now when I look back.
That's great. But what led you? I'm trying to go further back. Did you always know that you're interested in science in general? You mentioned, doing a degree in pharmacy. What did you want to become when you were, I don't know, a child?
When I was a child, I wanted to become a teacher.
Okay.
Still not completely out of the game, I
No.
would say. It's still possible. But then after graduating high school, I knew that I wanted to do something with natural sciences in whatever way or whatever flavor. The other possibility would have been medicine, but then I read up on it and I wanted to have a bit more chemistry because I really enjoyed chemistry in school. And then I got in contact with a pharmacist, which my family knows in my hometown, kind of. And he just borrowed me some books that he used during his studies. And I quickly screened through them and I saw a lot of chemistry there and I thought it's interesting. So I just decided to go for pharmacy. I was accepted and then I continued with that because I really enjoyed most of it, at least. Yeah. Not everything, but most of it was quite good. Yeah, and then after the pharmacy study, that's when I had to make the decision if I want to be in a public pharmacy. I had to make an internship as well. That's how the system kind of works in Germany. If you want to have your license as a pharmacist, you need to have a practical year afterwards. And 6 months of this practical year were in a public pharmacy. So I had to do this internship and I did it, but it was fun. But I knew that this is not the stuff I want to do for the next time, at least. Yeah. Then it was pretty clear that I would go for a PhD in more the scientific career, at least for now. And then it kind of developed.
That's great. Yeah, I was gonna ask how come you didn't become a pharmacist and stop there, but I guess you felt the need to develop molecules, to better understand molecules, and to really be at the forefront of research.
Yes, that was kind of how it went. It was still fun, but at least at that time I knew that this is not how I want to proceed.
That's you make a very interesting point, and I think it's important to highlight it. Is that you have to try out something first to then figure out if you like it or not.
Yes.
And a lot of I was talking at the conference at the GRC to a lot of people, and there is this among us, I want to say, and I'm included in that, this fear of failure or making the wrong move and quote unquote wasting time. But I don't think that should be viewed that way. Any detour you might take, yes, it might be a detour from your original goal, but going back on the track that you really want to go on with that added experience, it just changes you. So I think it's important to highlight if anybody is thinking about doing something that they're not sure about and you feel strongly about trying it out, go ahead. It's okay.
Yes, I'm usually not too good at that, I have to say.
I was not either, but yeah. That's why I wanted to make the point, because I think it's important for people to understand. And as you know, I quote unquote got out of the lab as the pandemic was hitting, and I went into multiple commercial roles, which are connected to science. But let's face it, I don't get to read as many papers as I would like to. I don't get to do as much science as I would like to. And I realized that I missed it. But at the same time, getting into those commercial roles, it was very difficult because people said, oh, you're a bench scientist. You can't, quote unquote, sell things. And I said, well, I'm not selling things. I'm providing support to scientists and reading the papers and telling them about the products and all of those things. And it allowed me to gain valuable experience that I think is, it's a detour, but I think it's also an eye-opening detour. That's when you realize what you actually really, really like. And you can also show yourself that you can do anything with a PhD.
That's also comforting to hear for my future.
Well, I'm glad. I think it was during the career panel we had that we were talking about the different career options and all of these things. And it's important to keep an open mind. You never know. All right. We've talked about frizzleds a little bit, about their importance in malignancies. There's 10 of them. What is the sequence homology between these? Because you mentioned the difficulty to specifically target one frizzle at a time.
Yes. So frizzles in general are very conserved, at least among the different species. There are differences, of course, but if you, for example, compare a human frizzled ape and the mouse frizzled ape, that's one of the examples that comes to my mind immediately. They are not too different. You can still recognize it as a frizzled ape. Among the 10 frizzles, I don't want to say something wrong. I don't know actually. I don't know how the sequence conservation is. There are some hallmarks kind of that you will always see in a frizzle, and as soon as you play with them, you get the same effect.
Okay.
So they all have this extracellular domain which binds the endogenous ligands, which is the CRD, the cysteine-rich domain. Not fully conserved, but these cysteines, for example, that's why it's called cysteine-rich domain. They are very conserved, and if you manipulate them, you will get a loss of function. They also have motifs which are conserved among the frizzles. But for the full sequence, I actually don't know the percentage. I would have to look it up.
I think, well, then I think for and this is very interesting too, because then it means that since the outside region is very conserved, hence the difficulty to develop small molecules or antibodies to differentiate between these. Although maybe, well, it depends on this. If it's a small molecule and it enters the receptor, then there must be some conserved regions anyway.
Definitely, and that's also always a problem that we run into with frizzles to achieve selectivity. That's right now, at least, it's still a goal, because of course, as we've said before, it's good to have selectivity, just to avoid any side effects, adverse drug reactions, maybe in the future. But that's definitely a problem.
It is. And there's 10 of them. It's not like there's 2 or 3, there's 10. So
yes.
Good luck doing. And I'm pretty sure we'll find a way to discriminate between these. But it's a matter of time and maybe methodologies.
Yes, but right now we still know that you can subdivide them in 4 clusters. On sequence homology, just to add up on your previous question, in those 2, it's usually 2 to 3 receptors per cluster, and those are very, very similar in many regions of the receptor. But still, you will find differences between them. Otherwise,
wow.
It's a bit of redundant.
Yes, absolutely. I think Mother Nature knows best when it comes to these things, and it's just fascinating to discover, the minutiae, the little differences that change the function, that change the expression. I think it's just beautiful. It's that's my GPCR-ness coming out there. So we talked a little bit about the difficulty of working with frizzled receptors. Then developing, for example, the bread-based biosensors to better understand their function and their biology. What do you think? And again, it's your opinion here that I'm interested in. We can speculate. If you had a magic wand and you can come up with 1 or 2 tools that you think would help better understand and really have this, I'm thinking here about a table that you had all the frizzles, and then you can have a full description of their function for each. What would that tool look like?
Oh, that's a very good question. I don't know how to design such a tool, but it would be great. There are two big questions now in the field. One question is wind frizzle selectivity, because we have 19 winds in mammals. We have 10 frizzles, and that doesn't really work. So it's definitely not a one-to-one. And we are now making the switch from overexpressed to endogenous systems, but still in an overexpressed system, if a wind is capable of interacting with a frizzle, we still don't know what it actually means for the endogenous system or for physiology. So that would be one tool that I would like to develop, just to have everything like a big toolbox where I can see, okay, this wind interacts with this frizzle in this context. That would be great. And the other question is kind of the mirror image on the intracellular side, like how frizzles interact with their transducers, and especially how they pick their transducer. I mean, we made some very good progress in answering these questions, at least partially. But still, that would be the second toolbox I would like to have, to see when does which frizzle interact with which transducer, and what is the consequence on the cellular level.
I think those are two very interesting questions. And you mentioned the 10 frizzles and the 19 winds. It kind of reminds me of the 20 chemokine receptors and the 50 chemokines.
Yes.
Which again is context dependent. We know that it's not one-to-one. And I think it's kind of, that's what it reminded me of.
Yeah, maybe not too far off, at least in terms of that.
Yeah. Although I have to say, compared to frizzles, maybe chemokine receptors are sometimes easier to study with the traditional tools we have.
Probably, yes. I've not worked with chemokines. I've only heard that they are tricky in many assays, but at least you can apply most of or some of the conventional tools. For us, it's really, if we want to look at something, we have to develop the tool first and to test it.
Wow.
That's maybe a difference.
Yeah. Well, most I want to say regular or typical chemokine receptors do induce beta-arrestin recruitment. They do transduce signaling through GI proteins. So you have all these tools. There is fluorescent chemokines that you can, although expensive, that you can get. Chemokines are very, very expensive to get. That was always the difficulty, but I think there's a lot of tools that we can use now. The difficulty remains again in the physiological setting, because these things are co-expressed. These things are in the cell, they're subjected to different chemokines, and there's other GPCRs in the cells. I think that complicates things a little bit, just a little bit.
Definitely.
And I do ask this from everyone, and I think the answer has always been yes, but I'm more interested into the why when it comes to thinking about GPCR as good drug targets. I think they're good drug targets just by looking at remembering just Fiona's talk at the conference. And I think a lot of speakers at the GRC had the slide, or at least they said it, that 30 to 40% of FDA approved drugs target GPCRs. But why do you think GPCRs are still good drug targets?
Well, I definitely think they are. I would totally agree with the majority of your guests, and also with you. I mean, it's always kind of the general introduction to GPCR papers that 40% roughly of FDA-approved drugs target GPCRs, but not all GPCRs are targeted yet. And I think there is a huge amount of GPCRs which are not targeted yet. I don't want to say that all GPCRs should be targeted or can be targeted in the end without any massive effects or side effects. But at least there is a huge potential, I would say, because GPCRs are involved in most physiological processes, and also in pathophysiological processes then. So I would say there is still a huge potential which we haven't discovered it. And even for other receptors like the opioid receptors, which have been hot for a while now, and they work in a way. They are analgesic, but of course, if you look at the side effects, there is still some room for improvement. So even the GPCRs, which are already targeted, still offer some room for exploration, for improvement, I would say.
I think so too. And I think it comes down to that complexity of having multiple GPCRs within the same cells, within the same tissues or the same areas. And the fact that we don't yet have the tools to study the whole effect on an organ or on the body. We see the consequences and side effects.
Yes.
But we don't yet, it's interesting. I think about this quite a lot, about the fact that cells are really magnificent and their ability to integrate multiple signals. And it's not one tuning, is multiple tuning buttons, and you're wondering, okay, who's doing what in the cell?
I agree.
And I think that's why. And I'm also wondering about, and this is again, this morning is open question morning. I'm wondering about olfactory receptors that we typically, which are very difficult to work with, and we know that they don't only exist, in the nose, they're everywhere. And I'm always wondering, like, what do these guys do exactly? And we don't have yet the tools to allow us to study them.
Yes, I agree, especially after the talk at the GRC.
Yeah.
I was always aware that they exist, but I also connected them more with olfaction. I knew that they were somewhere outside of the nose and the olfactory region.
Yeah.
But their effect is, or their expression is very, very interesting, I would say. They must be there for a reason. So that's, of course, a huge family, or a lot of families that we can explore.
Yeah, I thought so too. And that's the talk that popped into my mind as well. I think Ashishi's talk was really, really good. And I really like their approach about, generating these synthetic kind of mixed receptors that they were able to work with. So a lot of work to do, a lot of tools to develop.
Luckily.
Top 3 aha moments that you had as a scientist that, got you to where you are today.
That is a very tricky question. I'm not that long in the business, so I don't know if I have 3, but for me as a pharmacist, and it's also more or less the same answer that Hannes already gave in the podcast. For me, the first successful cloning was really an aha moment. I did not know that this is possible because this was not in the curriculum of our studies. Mm-hmm. So just being able to manipulate a receptor sequence how you want it to be, like attaching a luciferase to the N-terminus or to the C-terminus or somewhere else. I think this was the first big aha moment for me. The second one was then also, I've never worked on a confocal microscope like others did. Yeah. I've used a confocal microscope, but not for these ligand stimulation experiments, something. I've never done that. But also seeing the first result coming out of a plate reader that was successful, and kind of seeing the saturation binding curve, and also having a matching KD value, that was kind of an aha moment. And then also during my PhD, I would say there were some receptors which did not work for me. And it took me almost 3 years to get them working. And then after 3 years, when I saw, okay, this strategy now works, and that was probably the biggest aha moment, I would say. Yes.
I love it. You get to know those receptors. I always tell people that, every GPCR has their own personality, and you just have to get to know them a little bit, and then they might let you in on how they function. Obviously exaggerating, but I think one aha moment that transpired through our conversation here is you doing that 6-month internship in a public pharmacy. And I think that kind of made you realize, well, although I like this is not exactly what I want to do.
Yes, that's, if you want to describe this as an aha moment, I would agree.
Yeah, that was a career defining moment, because if you liked working in a pharmacy, we wouldn't be talking today.
That's true. That's true.
All right. Any advice for young scientists aspiring to contribute to the field? Anything?
Well, I think it's definitely good to be curious, and just if you if you know what you're working on, kind of, and then also just try to look a bit outside of the circle, which techniques are available on the market, kind of, or in the community. Maybe I can transfer these to my problems or to my project. I think this is an advice that I could give, if I may, in my state. And the other one is staying persistent. Really, don't give up when it doesn't work the first time. I mean, this is something that I had problems during my PhD project as well, as most people do, but now with the frizzles in the beginning, it was very frustrating because everything I touched did not work. Everything I tried to transfer from my previous work just did not work as I hoped or as I expected. But then just stick with it, try it again, try to modify it a bit. Maybe also, if nothing helps, try to start another project in the meantime, a smaller one, which gives you some results, and then you can always go back. Maybe you learn from the other project. But these are probably the two pieces of advice I could give.
Interestingly, you mentioned also the fact that before, you talked to Hannes before applying to the lab, and I think that goes to doing your homework and talking to people, and it's good to know where you're headed. It's good to talk to people who are in the lab, get a feel, and because it's important to be in a group where you feel comfortable, where you have the support of your PI, where you have the support of your peers, whether it's to do anything administrative when you arrive in your new country, new adopted country, as much as, telling you it's okay if it didn't work, let's try and think about it, or let's talk about it together.
Yes, that's definitely the case. And this is what helped me in the beginning as well. Gunnar is very supportive. He always told me kind of heads up when he saw that I'm a bit down. But then he's also very supportive when it works, of course. So, or if it works, so go that way. Or maybe you could try this. So that's a very good atmosphere that we have in the lab in general with both supervisor and my colleagues.
That's awesome. Okay, one last question.
Yes.
What next after your postdoc? Only if you're comfortable answering.
If I knew, I know that I want to stay in academia. Mm-hmm. I really like working in academia. As hard as it may be, but right now I would say that I want to stay. But I don't know in which function, in which position, which kind of position, and also in which country.
Yeah.
I'm German, so Germany is, of course, a possibility going back to Germany. Germany is also interesting in terms of GPCR research, but Sweden is as well. And Sweden is a very nice country. I enjoy it here. So both are possible. But academia is kind of the way or kind of the field where I see myself in, at least in the next few years.
Mm-hmm. And teaching, obviously.
And teaching, yes. And teaching.
Well, I think that's a great way to go about it. At least you know what you want. And this is by no means any advice, but I kind of giving it to myself retroactively, figure out what you don't want as well. That's also very important.
Yes.
So that you don't end up, if you know what you don't want, that's already a good thing.
Yes.
Amazing. All right. Thank you so much, Lukas. It was really amazing to chat with you. We're going to stop recording. And I forgot to mention this before we started. Don't go anywhere. We're going to let people wonder what we talk about right after. But it was really great chatting with you on the podcast. Thank you so much again for coming.
Thank you for having me. Thank you for the invitation. It was a pleasure.
Of course. Thank you. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest. We also want to say thank you to our Dr. GPCR team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners as well, Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem, so don't forget to join us today. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. With any questions or suggestions, feel free to email us at any time at hello@drgpcr.com. Until next time, stay safe.